Okay, anatomy and physiology students, I'm just going to quickly take you through our online Leech Neuron simulation lab. As we do this, we'll just see the equipment that gets used in the lab. I want to remind you this would be a pretty simple lab to do if we had just a little bit more time, and there's a couple of things that are complicated about it, but you can see you just need a dissection tray, a tank for the leeches, alcohol so they don't feel anything, a dissection microscope. The oscilloscope, we would just use a computer, some dissection instruments, and the complicated piece of equipment is this micromanipulator, which can be pretty expensive, and what it manipulates, which we have to make a tiny glass tube electrode. That can be a difficult process to master, where we take a piece of Pyrex tube, heat it up in a Bunsen burner, and stretch it out until it's microscopically thin, and then fill it with some saltwater solution so that it will serve as a recording electrode and conduct electricity. So, and then you have to put it in the micromanipulator once you expose the Leech nerve cord in order to be able to in order to carefully insert it into a neuron cell and then record the voltage difference from inside that cell and outside the cell. Remember, what we're seeing is the voltage difference due to the ions that are being pumped across the membrane. So how do we get to that point? We take the Leech tongs and we dip them in the water and pull the leech out and dip it in alcohol so it's anesthetized, and then we put it on the dissecting tray and we look at our dissecting tray and spread the leech out and we can then pin it down on the tray. So now we pin it down on the tray with the dissecting pins. We use our scissors to cut it open. What's amazing about a pretty primitive animal like this is when you cut it open, what you're going to see inside here is going to be mostly gonads. That with the ovaries with eggs, or the testes with sperm. And so there you can see that's all this animal really is a mobile factory for making more leeches with all that material inside there. And so we would just scoop all of that out. Just a moment. And so we take our probe and scoop all of that out of there. And now we're scooping the ovaries or eggs, sperm, ovaries, or testes out of the leech, and thereby exposing the nerve cord along its ventral surface. They actually have a ventral nerve cord. And so now we're going to look at it under the microscope and use a scalpel in order to cut open a little window in that nerve cord. And once we've done that, we can see the nervous tissue inside there. It's a little bit easier than to actually just get rid of the rest of the leech, and we can just cut out the body segment that we're interested in, and we have to flip it over and expose the nervous tissue on the other side. So we pin this down, and now we're going to look at it in the microscope again. And this will allow us to see exactly what is inside the leech nerve cord. Okay, so now we're looking inside the microscope again at that nervous tissue along the nerve cord, and we're going to bring our micromanipulator over, and now we can move that around. We're looking at the nervous tissue and try and stick our little glass electrode right into one of the nerve cells. And when we do that, in this case they're showing us that that cell is having an action potential. It probably wouldn't actually be beating like that, but it's just they're just showing us that it has an action potential. And then we can try and touch the outside of the animal and see if we get an action potential by using one of these instruments. So you'll see when we use something to touch the leech, it actually triggers an action potential. See it right there. You once again watch what happens when I touch the leech, and on the oscilloscope you'll see the action potential come up. One more time. Boom, there it is. You see the action potential on the oscilloscope. And so that tells us that it's probably a sensory neuron. The other thing we can do, because this is actually a little glass tube sitting in the cell, we can now, instead of using it as an electrode, we can inject some dye, fluorescent dye, into the cell, and then we can look at it under ultraviolet light, and we can see, sure enough, it has the cell body up to the side. It has one or two, but basically just one axon coming along here, suggesting that it's a very simple sensory cell. I don't care about you knowing the different kinds of leech neurons, but just the idea that that's a sensory cell. If we go back and probe another cell, then we might be able to find one that seems like more of motor cell, if we now touch the leech, you'll see, oh, no response. We're just not getting a response when we touch it. And now if we inject a dye and then turn on the ultraviolet light, we see we have a whole bunch of dendrites coming into the cell. It probably has an axon continuing off either this way, or one of these might be an axon. It's just not showing where it continues to, but a whole bunch of dendrites coming in, much we would expect from a motor cell. So we can see that and tell that fundamental difference between a sensory and a motor cell. So that's our little online Leech neuron simulator. Quick review of that. Thanks. Bye-bye.